So as Brian said, we'll be talking today about the regulations and ethics and how they relate to working with animals in research. So first we'll be discussing the importance of live animal models in research and education. So why do we need them? Often when you're talking to people outside the research field, they'll bring up questions like, why do we have to use live animals? We have other alternatives like computer simulation or cell culture. Why can't those be used instead? Or you might be asked, do the results we get from animal studies, can they really be applied to humans? We'll also talk about what we've learned so far from animal research, and I'll give a few examples of some research fields where animal models continue to play a large role. We'll talk about the difference between animal rights and animal welfare, and also the groups that oppose animal research. Another question that is often brought up when talking about ethics and animal research is, are these animals protected? So we're going to talk about the regulations and policies that are in place to protect the animals that are used in research and teaching. And then we'll also talk about your role and responsibilities as researchers in complying with those regulations. So there are several answers to the question, why do we need to use live animals in research? First, in order to develop treatments and cures for human diseases, we have to understand the basic physiology that we all share as vertebrate animals, as well as the pathophysiology of specific disease processes. Secondly, animals are necessary to provide an index of safety prior to work being conducted in humans. So the Nuremberg Code, which I don't know if you all done any human subjects protection, you'll probably talked about this. It was created in 1949 following the military tribunal trials of several German doctors who conducted unethical medical experiments during World War II. And the Nuremberg Code outlines 10 basic principles that must be observed in human subjects research in order to be considered ethical. And those principles include things like voluntary consent, avoiding injury and suffering, being able to terminate the experiment at any time, that sort of thing. And principle number 3 specifically addresses the importance of animal research. And that states, the experiment should be so designed and based on the results of animal experimentation and a knowledge of the natural history of the disease or other problem under study. The Declaration of Helsinki was first adopted by the World Medical Association at their General Assembly in Helsinki, Finland in 1964. And this really expanded upon the Nuremberg Code, and it too addresses the importance of preliminary animal research. And it states that medical research involving human subjects must conform to generally accepted scientific principles, be based on a thorough knowledge of the scientific literature, and adequate laboratory and animal experimentation. The third reason that we need to use live animal models is because the body's made up of multiple cells, tissues, organs, and they all have very complex interactions with one another. And those processes can really only be fully studied in an entire living organism. And then the use of animal models is not only important for research, it's important to train our next generation of scientists, as well as physicians, nurses, veterinarians. When you look at the actual numbers, you can see that the vast majority of the animals used in research are mice and rats. Only about less than 1% are dogs, cats, and non-human primates. This is because rodents are convenient, they're small, they reproduce quickly, they're easy to handle, they have short lifespans. Also, we have inbred strains of rodents which allow us to have more uniformity between studies. And then we'll go into more detail later about how rodent biology, behavior compares pretty well to humans. And then the remaining 4% is made up of all these other species here. And this graph in the right-hand corner, which is like very tiny writing, sorry about that. So it's comparing the number of animals used in research every year to those used for hunting, those killed by automobiles, and those consumed for food every year. And I had to double check this number for chickens because it's so astronomically high. And that is correct. There's over 9 billion chickens every year consumed for food. This is only the United States. So we really like our chicken here.
Wow.
One argument that's often made against the use of live animals is that we have these other alternative technologies, such as cell culture and computer simulations that could potentially take their place. And these alternatives are a very important part of the research discovery process, but unfortunately they can only take us so far down that path. So they actually make excellent preliminary screening tools for new drug treatments, for example. But again, they can never fully replicate all of the complex processes that are going on in the whole living organism. In addition, follow-up testing in animals is often required by law. You may have heard what's called the 2-animal rule, and the FDA typically requires that a new biologic product or drug be tested in more than one animal species before it can be tested in humans or approved for human use. So again, while in vitro models are important, at this time they don't have the power to completely replace animals. So another question that's asked is, can the results obtained from animals be applied to humans? And the answer is yes. There are many similarities between humans and animals, and I've listed some examples of some areas of study where it's very similar to those species listed. In addition, we have transgenic and even humanized rodents now, and this allows us to create specific human disease conditions in those animals. Transgenic zebrafish have also recently been generated, and they're really taking off in popularity as animal models for human diseases. The importance of these animal models is evident when you consider that nearly all medical advances were in some way shaped by animal research. Of the 105 Nobel Prizes awarded in physiology and medicine, 91 of those were directly dependent on animal research. This includes every prize awarded in the past 30 years. And in an additional 4 instances, those Nobel-winning discoveries were relying on data obtained from previous animal studies. These next 3 slides show some of the most important scientific discoveries of the past centuries, as well as the animal models that were used to perform those experiments. So all of our vaccines for animals and humans, organ transplants, imaging modalities such as CT, MRI, as well as treatments for a wide host of diseases, asthma, cancer, hepatitis, Parkinson's disease, HIV. So I presented a lot of historical data, but the research work to understand the underlying mechanisms of these disease processes and to find new and improved preventatives and treatments is ongoing. So we all know that obesity is a health epidemic in the Western world. In the United States, 64% of adults and 1/3 of all children are considered overweight. And this leads to further disease processes such as diabetes and heart disease. We do have several mouse and rat models that allow us to study the development of obesity and the causes of overeating and weight gain. This guy here is the Zucker obese rat, also sometimes called the Zucker fatty rat. He has a mutation in the leptin receptor gene and is characterized by obesity, overeating, the development of type 2 diabetes, and high triglycerides. Another field which has gained a lot of research focus lately is infectious disease and the role of these agents as bioterrorism agents. As I mentioned earlier, animals are a necessary component of the vaccine and treatment development process as dictated by the FDA. Tulane has played a large role in the understanding of the pathogenesis of HIV and the development of AIDS. We have an excellent monkey model of HIV and AIDS that was instrumental in the development of the current antiretroviral therapies, and it's still being used in the search for improved therapeutics as well as a potential vaccine for AIDS. And these are just a few examples of the many ongoing needs of animals in research. So next we'll talk about the difference between animal welfare and animal rights. So animal welfare is based on the concept that humans have a moral obligation to provide for the well-being of animals. Because we use animals for food, companionship, research, entertainment, then we have to make sure that those animals that are providing for us are treated humanely. This is different from the concept of animal rights, which is the viewpoint that animals have the same rights as human beings. People who believe in the rights of animals believe that we should not be allowed to own or use animals for any reason, including food, clothing, entertainment, research. And some individuals actually carry this belief so far that they think that we don't have the right to use animals for police or disaster recovery work, or even to own pets. There are several organizations for people who identify with animal rights. Probably the most famous that everyone's heard of is PETA, People for Ethical Treatment of Animals. Their director and co-founder, Ingrid Newkirk, has been quoted as saying, "Even if animal research resulted in a cure for AIDS, we'd still be against it," as well as other inflammatory remarks, such as supporting arson against research labs. Now, the majority of animal rights activists pursue their campaigns legally and peacefully with protests and advertising campaigns, and that's their legal right to do so. This is in contrast to animal rights extremist groups who advocate violence and terrorism to achieve their goals, and the FBI does consider these groups to be terrorist organizations. Examples of a couple of these extremist groups who are active in the United States include SHAC, which stands for Stop Huntington Animal Cruelty, and ALF, which is the Animal Liberation Front. And their spokespersons have stated that they consider violence, including murder, to be necessary and justifiable. Luckily, in 2006, the Animal Enterprise Terrorism Act was passed, which expanded protection for academic and commercial institutions that use and sell animals, and increased the existing penalties against animal rights extremist groups. And it has led to the successful conviction and imprisonment of several individuals who are targeting researchers. So next, I will talk about how the concept of animal welfare as it relates to research animals has evolved over time in the US. So in the 1940s and 1950s is when we first started seeing this increased concern by the public about the use of animals in research, and specifically as it related to pets. People were afraid that pet dogs and cats were going to be taken and used for research experiments. In 1954, the University's Federation for Animal Welfare commissioned a systematic study of laboratory techniques in an ethical context. And the results of that study were published by Drs. Russell and Birch, that's those handsome fellows over there, in their publication, The Principles of Humane Experimental Technique. And this document was the first to outline the concept of the 3 R's, which we're going to talk about more on the next slide. Then in 1965, the Association for the Accreditation of Laboratory Animal Care, also known as ALAC, was founded. ALAC is now an international organization, and they are a private, nonprofit accrediting body, and they promote the humane treatment of animals used in research through this voluntary assessment and accreditation program. Tulane actually has the distinction of being one of the first institutions to receive ALAC accreditation, and we have maintained that accreditation for over 40 years. We're really proud about that. So the 3 R's are an important concept that still make up the backbone of animal welfare today. They stand for replacement of animals with non-animal models, such as cell culture and computer simulation, or with less sentient animals, such as Drosophila flies or flatworms, for example. Reduction of the number of animals to the minimum number possible that allows us to get statistically significant results. And refinement of our techniques to reduce pain and distress whenever possible. And federal regulations require that every research institution in the U.S. practice the 3 R's. So one of those federal regulations is the Animal Welfare Act, which was first signed into law as the Laboratory Animal Welfare Act in 1966. It's enforced by the United States Department of Agriculture, and it's been amended several times to expand protections for laboratory as well as other animals. In 1985, the Health Research Extension Act was passed, and that is enforced by the NIH Office of Laboratory Animal Welfare, and it applies to all public health service funded research. So those federal regulations were put into place to ensure that the animals being used in research and teaching, as well as other uses, were being treated humanely. In addition, the Public Health Service Policy requires that we follow a document called the Guide for the Care and Use of Laboratory Animals. This is actually a book. It goes into very specific detail on how the animals need to be cared for, down to the temperature in the room, the lighting, and even how many air changes per hour the HVAC system has to have, as well as the size of cages the animals need to have. The PHS policy also requires that we follow the AVMA panel on euthanasia, which outlines the humane euthanasia techniques for each species. Both the Animal Welfare Act and the PHS policy are broken down into sections, and they describe the requirements for adequate veterinary care, appropriate housing, feeding, handling, sanitation, ventilation, as well as monitoring of the animals. Each institution is also required to have a documented emergency plan. And every proposed research study using animals has to undergo review and approval by an institutional animal care and use committee, which we'll talk about more later. And if an investigator is found to be non-compliant, either with the regulations or with their protocol, then Tulane is required to report that individual to the federal government as well as their funding agency. Because Tulane receives money from the federal government, we're required to file an animal welfare assurance with the NIH. Basically says we're going to follow the law. And the NIH is allowed to come out and inspect us at any time, unannounced, although they typically won't show up unless there's a serious problem. Tulane is also a registered research laboratory with the USDA, and the USDA does come out every year unannounced to inspect us. And since we are accredited by ALAC, they come out to visit us every 3 years. And ALAC standards are very high, actually higher than what's required under the minimum by law. They do a thorough review of every aspect of our animal care program, including our facilities, the iCook, occupational health program, and the veterinary care of the animals. So now that you're familiar with the regulations, we'll talk about what your role and responsibilities are as the researcher in complying with those regulations. And it's not just your responsibility as researchers or my responsibility as animal care staff to make sure that we have a strong program of animal welfare. It's really a team approach that requires cooperation from the investigator, the IACUC, animal care, as well as the leadership here at Tulane. So we'll start with the role of the leadership, and specifically the individual called the institutional official. That individual plays a very important role. They're typically a very high-ranking official at the institution, often the CEO or president or vice president. Our institutional official here at Tulane is our Vice President of Research, Dr. Laura Levy. Her job is to provide leadership, disseminate all of the policies, and to set the tone of the research here at Tulane. The IO also has to have the power to allocate resources to maintain quality animal care and high-level research. They are the one who appoints members to the IACUC, and they're also the one that provides an annual report to the USDA and OLA every year. We're actually very lucky here at Tulane. We have an excellent institutional official who really believes in the need for high-quality animal research, and she actually uses animals in her own research, so she understands the unique needs and challenges that y'all face as animal researchers. Each institution is also required to have an institutional animal care and use committee as part of the animal welfare program. The IACUC is responsible for upholding the standards and the regulations as well as our institutional policies, and no research can proceed without IACUC approval. Each IACUC committee is required by law to have a veterinarian, a scientist, a non-scientist, and a member of the public who is not affiliated with Tulane. When reviewing the IACUC protocol, the committee is looking for a justification for the use of animals. They want to see that all the individuals listed are appropriately trained or have experience for the roles that they're going to be doing, that the investigator has considered alternatives to the use of animals as well as to the use of painful techniques, and they want to see how pain and distress is going to be alleviated. So again, this all goes back to the 3 R's that we talked about earlier. In addition to reviewing protocols, the IACUC also reviews the institution's entire animal care program every 6 months. This includes inspecting every room on campus where animals are housed or used, at least every 6 months. They're also the ones who investigate any concerns that are brought to them about animals on campus, and in the event that an investigator is found to be non-compliant, either with their protocol or with the regulations, they have the power to suspend that investigator's research and report them to the federal government. The next member of our team is the animal care staff. This includes our veterinarians, veterinary technicians, animal care staff, cage wash, maintenance people, everyone that keeps the animals healthy, happy, and the facility running smoothly. Each institution is required by law to have an attending veterinarian who is a member of the IACUC and is ultimately responsible for the care and health of the animals. In addition to clinical duties, the attending veterinarian is responsible for the management of the animal facility, and they provide training and technical assistance to anyone who works with the animals. And this training can be a range of activities that you see there listed. Another service that I provide to investigators as the attending veterinarian is assisting with IACUC protocol preparation. The attending veterinarian also has the power to halt any research activity that they feel that the animal's welfare is at risk or that the work is being performed out of compliance with the protocol. And it's my responsibility as the attending veterinarian to report any animal concerns or noncompliance to the IACUC. My staff is responsible for the daily husbandry, socialization, and enrichment of the animals, as well as the management of the facility. Members of the animal care staff often serve as members of the IACUC, and really they are on the front line when it comes to animal welfare. The majority of the non-compliance or animal welfare concerns that are reported to the IACUC are first reported by the animal care staff. And that's because they're the ones who look at the animals every day. They're the first to know when something looks unusual or if an animal's unwell. So our staff here at Tulane, we take care of your animals 7 days a week, 365 days a year, including weekends and holidays. We're also essential personnel. Members of my staff stay on site in the event of emergency closure. And there's also always a veterinarian on call during weekends and holidays. So now finally, what are your responsibilities as investigators in maintaining animal welfare? First, you have to obtain IACUC approval prior to beginning work with the animals. You have to obtain your animals through approved means. This is usually through my office, unless you're working out in the field with the wild animals. And you cannot make any changes to your experiments until you've submitted an amendment to your IACUC protocol and had those changes approved by the IACUC. It's very important. In addition, it's your responsibility to cooperate with the IACUC, provide any information they request regarding your experiments or the animals. As I mentioned earlier, the IACUC does have the power to suspend an investigator's research. So they would only do that if they felt that there was serious or continuing non-compliance that was going on that would threaten the welfare of the animals. But should they suspend your research, you're required by law to comply with that. It's also very important that you do not allow your protocol to expire, that you still have animals present in the facility. The federal government does not allow animal research to occur on an expired protocol. And our Tulane IACUC office is actually very good about sending out multiple notices far in advance of your protocol expiring. But you have to remember, don't wait till the last minute. When you get that taken care of right away. Because you have to factor in the time it's going to take for the IACUC to review that new protocol and get it approved.
How long in advance should someone?
The IACUC office sends out a 120-day notice, and then a 90-day notice, 60, 30-day notice. So they give lots of heads up that your protocol is about to expire, and I would give at least a month for the IACUC to be able to review, because they may request changes to be made to the new protocol, and you need to factor in that time it's going to take to do that. If your protocol does expire before the new one's approved, we have to transfer those animals over to my holding protocol. The investigator's access is taken away because, again, you can't do any research on animals on an expired protocol. And the NIH and other funding agencies will also not allow their funds to be used to pay for animals if they're on an expired protocol. We actually did have an instance a few years ago of an investigator who let their protocol expire for several months, and that individual had to find an alternate means to pay for the per diem for their animals because their NIH grant would not allow him to pay for them. You're also responsible for the staff and students in your lab that are working with the animals. So you have to ensure that anyone who's contacting live animals is appropriately trained and that they've been added to the IACUC protocol. You're also responsible for the animal records, and everything has to be documented that you do to the animal, either in your laboratory notebook or on another form. And those records have to be up to date, complete, and available for inspection at all times, either by the IACUC or by our regulatory bodies that come and inspect us. And you're also responsible for the safety of the personnel in your lab, making sure that they've enrolled in the Tulane Occupational Health Program. You also have a responsibility to report any concerns, either that you have or that are reported to you regarding animals. So if any of y'all ever have a concern about animals, you're encouraged to contact any of these individuals. We have their contact information on our website, also on Brian's website, and posted throughout the animal facility. It's very easy to find. Your identity can remain anonymous if you wish, and if you are a Tulane employee, then your job is also protected. Another important investigator responsibility is to ensure that euthanasia of the animals is performed in a humane manner. And it's our responsibility to ensure that euthanasia is performed with respect and as pain-free and stress-free as possible to the animals. So the method of euthanasia must be consistent with the recommendations in the AVMA guidelines for the euthanasia of animals, and that method must be approved by the IACUC. So you have to ensure that the euthanasia method you're using is described in your approved IACUC protocol. You have to make sure that lab personnel are appropriately trained and proficient in that euthanasia method. And this is another important one. You need to confirm that the animals are actually dead after you perform the euthanasia. This is especially important when doing the CO2 euthanasia, which is the most common form of euthanasia for rodents. This is why we have a Tulane policy that requires a secondary physical method of euthanasia following the CO2 method. And examples of a secondary physical method would be cervical dislocation or cutting open the chest, for example. And just like any other change to the experiment, you can't make any changes to the euthanasia method without IACUC approval through the amendment process. And my office is always very happy to assist anybody that has any questions or needs any help with euthanasia, because we want to make sure that y'all are doing it correctly. So that's all I have. I'd like to thank all these people who assisted with the seminar or contributed materials for these slides. And then if you want any more information on any of the topics we covered, these are really great resources. So does anyone have any questions? All right, well, thanks for coming. Appreciate it.